In 1958, there was a book published. The title was Eat Fat and Grow Slim. In 1977, the US released the new dietary goals, and they created that pyramid saying that you should cover the calorie intake mainly with carbohydrate, and on the top of the pyramid, they put the fat. From the 90s, at each age, everybody and the body weight is started to growing and still growing. The question is there any correlation or is there any link between that three independent facts? I would say that the link is the deuterium. So I do not need to explain that slide. It's clear. We talk about 150 ppm, and we know that we are drinking 3 different water molecules when we drink a glass of water. I guess the big difficulties was for 60 years, everybody think about that the ratio is 6,600 to 1 between the hydrogen and deuterium, and that was the reason that nobody investigated the possible role of deuterium. But when we explained the D level in millimole, it is clear that the D level is 6 times higher in the calcium, 10 times higher in the magnesium. And for the MDs, they know that 2 millimole drops in potassium concentration that can cause the death of the patient. So the lives very strictly regulate the calcium, magnesium, and 100 other parameters in the blood. And the deuterium is there, and it wasn't investigated. So let's see what happened when we modify the D concentration and what will happen with the metabolism. So we made the rats diabetic. It means we killed the Langerhans cells, which is developing and secreting the insulin level. And after making the rats diabetic, in the very first experiment, we checked whether the 25 ppm can modify the blood glucose level. When the rats did not receive insulin, that independently what was the D concentration of drinking water, the blood sugar level was high. But when the rats received twice 1 unit insulin, then the 25 ppm reduced the blood sugar level. So we were happy that the D concentration reduced the blood sugar level. The next experiment we used 25 ppm, 75, 105, and 125 ppm. And the point was that not the lowest D concentration reduced the best way the blood glucose level. So the 125 ppm was better than the 25 ppm. So we made another experiment. In that experiment, we changed the D level by 5 ppm, starting from 125 and go up to 150. And this is the summary of the rat experiment, which shows clear that the best dose to reduce the blood sugar level of the rats is between 125 ppm and 140 ppm. It's very important. It shows that this is not only depends on the D concentration. So here must be something that the best dose is not the 25. And that was true for the fructosamine, which shows the D level, blood glucose level, the average of last couple of months or weeks. And that was true for the hemoglobin A1C. So when a diabetic patient next week goes to the MD and want to show good results, when they check the hemoglobin A1C, that will be upper because in the last 2 months they did not keep the diet. So that, again, proved that this is the best dose for the patients, for the rats. What is the mechanism? We know that finally the GLUT4 protein responsible to pick up the glucose from the bloodstream. The GLUT4 typically is a cytosol. When somebody eats the food, the blood glucose level is increasing, that the pancreas recognize it and start to eliminate and send the insulin to the blood system. The insulin binds to the receptor, and that triggers a signal system, and that signal stimulates the translocation of the GLUT4 from the cytosol to the membrane. That is the basic mechanism. So to check it whether it's true or not, we tested what is the GLUT4 concentration in the muscle membrane of the rats. When the rats were healthy, independently whether they consumed 25 ppm or 155 ppm, the GLUT4 level was high in the membrane. But in the rat with diabetes, the lowest GLUT4 level was at 150 ppm and the highest was at 125 ppm. So from this experiment, we can conclude that the DDW can reduce the glucose concentration in the diabetic rats, but only in the presence of the insulin. And the data showed that the DDW stimulates the GLUT4 translocation from the cytosol to the membrane. And that is the optimal concentration. If we had an experiment with the rats, let's see what will happen with the humans. So we had an experiment, a phase 2 clinical trial. We enrolled 30 patients. Everybody consumed 1.5 liter per day with the water 104 ppm. And that lasted for 90 days. We calculated the deconcentration from the blood. At the beginning, the average blood deconcentration was 147 ppm. At the end of the trial, after 90 days, it was 133 ppm. We followed a couple of parameters in the blood, which was very, very important that we could prove that consuming 104 ppm DW that significantly reduced the sodium concentration in the blood. That was the only parameter which were reduced out of 30 patients, 29 out of 30 patients. And the good news that the consumption of DW can increase the so-called good cholesterol level with about 30 ppm. So the HDL went up to 1.34 millimole. The other good news was that the blood counts somewhere was better and increased, and that should be one reason that again, when the cancer patients consume DW, that somehow we can prevent the blood system. We checked whether consuming DW can modify the fasting glucose level. So at the beginning, it was 6.07. 3 months later, it was 5.74. Unfortunately, we can see a peak at 60 days. So we can explain that, I hope. The enrollment of the patients enrollment started in October. So that was December, that was Christmas time. We believe that people stay home, eat more, so that increased the fasting glucose level. Out of 30% of these 30 patients, we could see the reduction of the insulin resistance. So in 11 patients, the glucose uptake went up from 6.97 milligram per body weight per minute up to 8 milligram, 8.64 milligram per body weight per minute, which is very good news. If we talk about within 3 months, we can reduce the insulin resistance 30% of the population. What is the water source for humans? The one water source is when we drink the water. The other water source, which is made by the mitochondria. So we know that typically 10–15% of the daily water need is produced by the mitochondria. But what kind of water can produce the mitochondria? From the fat, the mitochondria can produce 100 fat, 107 grams H2O, and you can see the carbohydrate and the protein produce also less water. But the question is, what is the deconcentration of the metabolic water which is made by the mitochondria? And in the metabolic water, the hydrogen source is coming from the carbon source. So when we are eating carbohydrates, flour, sugar that produce metabolic water with close to 150, 146 ppm. But when the people eating fat, goose fat or lard, that is very, very depleted. And the final deconcentration of the metabolic water depends on the composition of the diet. The more carbohydrate, closer to the deconcentration. So if we compare the pyramid and we talk about what was suggested in 1977 by the US dietary, that produce about 141 ppm metabolic water, but when we talk about the ketogenic diet, that produce about 121 ppm metabolic water, which means we can get 20 ppm difference depending on the diet. So and the good news that in the United States, they turned up, changed the pyramid, so now the bread, carbohydrates at the bottom, and the fat and the meat goes up to the pyramid. If we go back, eat fat means those who are eating fat, they produce lower deconcentration by the mitochondria, and the average D level of the patient is much lower. And that could be close to the deconcentration, but we proved in the rat study that for the insulin signal system, the lower deconcentration preferred. So the whole population worldwide is living in not the best deconcentration. We are very close to the 150 ppm, and the life wasn't planned for that deconcentration. What about with the cell cycle and gene expression? We know that the cell cycle is a very, very complex system. It takes about 24 hours. The cells are staying in the G1 phase. And when they decide they are ready to move and go to the S phase, they disrupt the whole structure, and at the end of the cycle, they put everything together. So that requires very precise timing, switching on, switching off genes, changing everything in the cell, how the cells can manage it. And what is the point when the cell decide moving from G1 to S phase? Never we start the cell moving to S phase when the cells are not sure about in a G1 that they can complete the whole cycle. So this is the reason if we find that point, then we can cure cancer, because then we can intervene and we can modulate that whole system. This is how the drug development is moving. It's amazing how much we know about the signaling system in the cell. And I never want to learn these numbers and these letters, HKT, there are MOP, kinase, wherever. This is the target of the drug development of the pharma industry. And we know the data and we know the results. And I do not say that we do not need combine the DW with the existing therapy, but I say moving on forward, that will not solve our problems. So the extragen system is a system when we do not manipulate with the cells. The cells are growing on a gold layer, and the only thing we follow is the changing of the impedance of this gold layer. So that allows us to follow very precisely what can be caused by changing the deconcentration. So we prepared media with 150 ppm and in the lowering, and that shows that there was quite strong correlation. The lower was the deconcentration, the lower was the growth rate, or at 40 ppm, we completely stopped the cell growth. And when increasing the deconcentration of the 200, 300, 500 ppm, we could prove that somehow that enhanced and stimulated the cell growth. And when we first time, and one more experiment, here we checked how sensitive the system, how sensitive the cells for the deconcentration. So here we change the deconcentration in every 8 hours by 0 ppm, 1 ppm, 2.5, and 5 ppm. And as you see that the red line is different from the green line, which means that the cells recognize that 1 ppm decrease, and that finally reduce a lower growth rate. So when we explained that, and we published with Jancsó Gábor and the first co-authors, how could we explain our data that the lower D level reduced the growth rate and the higher D level stimulated the growth of cells? It was clear in the 80s that when a growth hormone binds to the membrane, that stimulates the sodium hydrogen antiport system, and that causes a pH shift. And it was clear that somehow part of the signal that the cell will grow. And that was interesting paper published in Nature, that they took out the ATPase gene from the yeast, and that gene was used to transform animal cell line. They could prove that the ATPase working in the membrane, transferring the hydrogen out of the membrane, but everybody was surprised that first that cell line caused tumorous type in vitro and caused tumor in nude mice. So one single gene, ATPase, make that cells cancerous. And how can we explain that when we are talking about 200 genes, which has a key role in the cancer development? So we explain that. When the growth hormone binds to the membrane and that stimulates the antiport system, that will prefer to eliminate the hydrogen, which leads a higher DH ratio in the cell. And we concluded that this is the signal for the cell. The cells measuring the DH ratio, and depending on what's happened with the DH ratio, they turn off genes and regulate the metabolism. So when we use deuterium-depleted water, the hydrogen antiport system is activated, but the cells cannot lift up the DH ratio, or it takes longer time. And we know that, Roman Zuber proved, changing the D level, that will trigger radicals, that radicals disrupt the system, trigger the apoptosis. So when a growth hormone binds to the membrane, stimulate that, at the same time there's a decrease in D level, that can disrupt the whole metabolism. And the good point is that there is a paper independently published from our research that the ATPase do not accept the deuterium. So there is a very, very strong discrimination between the hydrogen and deuterium, and this is how it can work. So they can separate these two isotopes. Albert Szent-Györgyi said at the 70s that you are not able to regulate the cell growth with one light. So if we say that the sodium hydrogen antiport would be the green light giving to the cell, we need a red light. And the red light would be the mitochondria that produce metabolic water. And as earlier I said, in healthy cells, the mitochondria and the sodium antiport is imbalanced. The sodium antiport is working, is able to increase the DH ratio, but a properly working mitochondria producing deuterium-depleted metabolic water. So they are playing a game, whether they can increase the DH ratio to the threshold or not, but they are balanced. And it's amazing that it's well known for 100 years that 90% of the cancer cells do not have a properly working mitochondria. So damaged mitochondria, no deuterium-depleted metabolic water production. So no balance, which would works again to the sodium hydrogen antiport, which is going to increase the DH ratio. So because the mitochondria is damaged, the signal is coming, very easily can lift up the DH ratio threshold, and this is the reason that finally the cell will enter the S phase and multiply. How can we prove that the mitochondria has a critical role in that game? So we replaced the drinking water of these laying hens for 25 ppm, and we started to collect the eggs week by weeks. And then we separated the yolk and the white we take out the water from the yolk and the white. And the D level of the water in the yolk and the white reduced in the same way. And you can see after 2 weeks or after 20 days, we reached the equilibrium. At the same time, we could see that there was some decrease in the deconcentration of the white, but there was a drastic decrease in the deconcentration of the yolk, which can consider as a fat. That yolk was added to the mice food, and we checked the survival of the mice. And we found that those mice, the control, after transplanting the tumor, they died. Those mice whose food was supported and supplied with deuterium-depleted yolk lived somewhat longer, but those mice who parallel with eating that deuterium-depleted food with yolk, drink DW, they lived even longer. So in that case, when the mice eating deuterium-depleted fat, that could increase the lifespan because that mice produce deuterium-depleted, much more depleted metabolic water. So we say that the mitochondria communicate with the cytosol, and the activation transport system prefers eliminate the LITER isotope, and that will cause a higher DH ratio. The healthy cells in the mitochondria produce deuterium-depleted metabolic water, which is a key role to control the cell growth. And in the cancer cells, as I said, no deuterium-depleted metabolic water production. This is the reason that finally the cell can enter the ASH phase. And we go back to the gene expression study. The nanosync technology is good to, we are able to count one by one how many copies was made from different genes using that technology. So in that study, we simultaneously tested 236 cancer-related genes and 536 kinase genes. And we used, as Csonka Gábor mentioned earlier, 40, 80 ppm normal water and 300 ppm. When we published our very first paper, I used a much simplified way to eliminate the bias. So we evaluated only those genes when the changes in the gene expression was higher than 30% and when the copy number was higher than 30. So based upon, we could see that finally we evaluated 124 genes, cancer-related genes, and 135 kinase genes, where the changes were in the expression 30% and the copy number over 30. Theoretically, we have nine way. The one way is that the deuterium level do not modify any gene expression. One possibility that DW downregulate genes, but enriched water do not modify, and vice versa. But the point was, this is what we received. So finally, out of 135 kinase gene, we have only one, which was downregulated in deuterium-depleted media, but 134 was upregulated. And we had something similar with the cancer-related genes. Only one gene was upregulated in DW and deuterium-enriched, 5 genes downregulated in deuterium-depleted, but all the others was upregulated. So finally, the whole genome was sensitive and regulated by the DH ratio, and they move for the higher DH ratio. So this is the conclusion. The deuterium to hydrogen ratio is primarily regulate the initiation of the cell cycle. The gene expression, the gene activation is the secondary response that regulates the progression through the S phase, G2, and M phase. So everything we say depends on the DH ratio. That submolecular changes finally is translating to the molecular level, and then the molecular level doing the gene expression, synthesize the proteins, and the proteins finally complete the job. Synthesize the DNA, synthesize the membranes, and everything. So we say that there is a submolecular regulatory system, and our aim is and target to develop drugs which can intervene in that submolecular level. I have a long list to say thank you, first of all, to my wife. And without her support, we would be nowhere. Thank you, Jancsó Gábor and Jákli György, the first two scientists who supported me 35 years ago. Without their help and measuring the D level of the water I prepared in my garage at home, I would be nowhere. Of course, thank you, László Boros, thank you, András Pap, István Forris, and all the others who take part in that long journey. Thank you.
Thank you very much.